This is question 3 of paper 1 on the 2017 A2 paper. It's about transport across membranes, so therefore it's great for AS and A2 students. Quickly reading through the question, sodium ions from salt are absorbed by cells lining the gut. Some of these cells have membranes with a carrier protein called NHE3. NHE3 actively transports one sodium ion into the cell in exchange for one proton hydrogen ion out of the cell. Use your knowledge of transport across cell membranes to suggest how NHE3 does this. So the stem of this question tells us where it wants us to go with it. It talks about a carrier protein, and it talks about actively transporting one sodium ion in exchange for one hydrogen ion. It's co-transport because it's moving two things. However, it's active transport because it tells you it actively transports a sodium ion. Now, although it's active transport, we don't need to start talking about diffusion gradients. All we're talking about is how the protein actually works. So we're going to say that this is co-transport using active transport, i.e. the hydrolysis of ATP. And just like with the sodium-potassium pump, what we have is a sodium ion and a proton, i.e. a hydrogen ion, binding to the protein. This causes a shape change and moves the ions across the membrane. It would be the same answer if you were asked about the sodium-potassium pump. So there were 4 possible marks for this 3-mark question. So my top tip for this question would be to stick to the stem of the question and don't just write everything you know about why ions would move. That is not what the question is asking. The next part of the question is a lot to read. What I would do is I would use a pen and I would highlight things as you worked your way through it. That way, when you flick your eyes back to the question, you'll go back to the main points. You'll also feel like you've read it properly. So let's have a quick read through. Scientists investigated the use of a drug called tenapanor to reduce salt absorption in the gut. That drug inhibits the carrier protein NHE3. So far, that's the only bit of the question I need to highlight, that the drug, doesn't matter what it's called, doesn't matter I've never heard of it, but it inhibits the carrier protein NHE3. Okay, then we've got 2 groups of rats. One group was given the drug and the other group wasn't. Then 1 hour after the treatment, the scientists removed the gut contents of the rats and immediately weighed them, and there's our results in the table. Scientists carried out a statistical test to see whether the difference in the means was significant. They calculated a p-value of less than 0.05. They concluded that the drug did reduce salt absorption in the gut. Use all the information provided and your knowledge of water potential to explain how they reached this conclusion. So they've told us in the stem of the question that the p-value was less than 0.05. It doesn't matter what statistical test that they've used. We don't have to worry about that. They've done the work for us. All we need to do is interpret the value of p. And that's where we return to our mantra that we've been learning, where we use the words probability and chance. We're going to say that there is less than a 5% probability that the difference in results is due to chance. And that in turn tells us that there is a significant difference between A and B. So now we've used all our information on the paper, and it asks us to use our own knowledge of water potential to explain how they reached the conclusion. So if we're reducing salt absorption from the gut, then more salt is going to remain in the gut. That's going to lower the water potential, so less water is going to be absorbed into cells by osmosis, because the water potential is going to be lower inside the gut than in the cells. So where were the marks? Well, you got 1 mark for mentioning in the sentence, appropriately, that there is less than 5% probability that the results are due to chance. So you needed both the words probability and chance. You did not get the mark if you said there was 5% probability, because that's very, very different. The second mark was for saying that because of that, there is a significant difference between the results. The third mark for talking about lowering the water potential if the salt remained in the gut, and the 4th mark for mentioning the movement of water, which was by osmosis. Okay, so top tip for this question, remember, don't say 5% probability, say less than 5% probability, and stick to the mantra There was less than a 5% probability that the results were due to chance. Don't start talking about not due to chance or 95% probability. Just stick to the plan. If you read the examiner's report, it will tell you that when students did actually try to attempt this part of the question, talking about the statistical tests, very few of them completed it successfully because they went off track. They need to stay on the mantra, stick with it. Okay, and the last part of the question, high absorption of salt from the diet can result in a higher than normal concentration of salt in the blood plasma entering the capillaries. This can lead to a buildup of tissue fluid. Explain how. Interestingly, according to the examiner's report, only 15% of students managed to gain both marks on this question, which is crazy. The first thing that we're going to say is that a higher salt content results in a lower water potential of the tissue fluid, because don't forget, salt, or the sodium ions if you like, are small enough that they're going to be forced out from the capillary into the tissue fluid. So if this is the case, we've got less water returning to the capillary by osmosis at the venule end of the capillary bed, because there's a lower water potential there, so it's holding on to the water. And remember, it's water that would return to the capillary by osmosis. You can't talk about tissue fluid returning to the capillary by osmosis because it's not tissue fluid by then, and osmosis is only the movement of water. Alternatively, we could have come from the other end of the capillary bed. We could have talked about the higher salt resulting in a higher blood pressure, so more fluid was forced out at the capillary end of the arteriole. Remember the structure of the capillary bed where we get the arteriole splitting into many capillaries and then reforming at the venule end. Okay, so my top tip for this question is to remember that it's water that's reabsorbed back into the capillary and not tissue fluid. Always to mention a lower water potential and not just a low water potential, and less water as opposed to water. I do hope you found the video useful. Please do leave me a comment.